Right now, your liver is actively diffusing a microscopic toxic bomb. When a patient presents to the emergency room suffering from acute acetaminophen-induced hepatic necrosis, their internal chemical defenses have been saturated and broken. At the exact same moment, the keratin structures of that same patient, their skin, hair, and nails, remain resilient, maintaining physical elasticity and a strict waterproof barrier against the outside environment. Both the internal toxic collapse and the external physical armor are governed by the exact same highly reactive molecule, a single sulfur-containing amino acid called L-Cysteine. Evolution selected this molecule during the transition to an oxygen-rich environment. Because oxygen is destructive to cellular integrity, early life required a reversible biological switch to manage systemic oxidative stress. To understand how human physiology survives in a harsh oxidative world, we must map this molecule's specific structural geometry, its restricted distribution across the body, the pathologies that arise when its transport fails, and the logistical challenges of measuring it in a clinical laboratory. L-cysteine is a precisely controlled biological explosive. It is dangerous to leave freely circulating, but essential to preventing structural collapse. We begin with the physical chemistry. L-cysteine is the only standard protein-building amino acid that features a terminal reactive thiol group, a sulfur atom bonded to a hydrogen atom at the edge of its carbon framework. Sulfur is a relatively large atom. Because of its size, its outer electrons are loosely held and sensitive to the electrical environment. In chemistry, this makes the atom polarizable. It is chemically desperate to share and swap electrons with nearby structures. When two separate cysteine molecules enter an oxidizing environment, they shed their hydrogen atoms and link their respective sulfur atoms directly to one another. This linkage forms a covalent disulfide bond. The thermodynamics of this specific chemical bond are significant. Creating a single disulfide bond releases approximately 250 kilojoules per mole of energy. This release of energy locks the three-dimensional structure of proteins into place. It operates like pouring concrete around steel rebar, giving our extracellular structures the mechanical rigidity to resist physical pressure without unfolding. The body restricts free cysteine, locking it into two guarded vaults. The integumentary vault holds 40%, anchoring sulfur inside keratin strands for physical armor. The intracellular glutathione pool holds 45% inside nucleated cells, acting as the raw material for internal chemical defenses. That leaves a very small percentage in the vascular fluid phase. Free circulating plasma cysteine is strictly capped at under 25 micromoles per liter. By keeping free cysteine scarce in the bloodstream, biology prevents circulating proteins from randomly supergluing themselves to one another and aggregating into massive, non-functional clumps. Inside the cell, cysteine is the rate-limiting bottleneck for synthesizing glutathione, a 3-amino acid peptide that acts as the master intracellular antioxidant. The cell can only build as much glutathione as it has cysteine available. This supply determines cellular survival. Normal metabolism continuously produces reactive oxygen species, free radicals missing an electron. Without glutathione to neutralize them by donating an electron, these free radicals violently attack the cell's lipid membrane in a process known as lipid peroxidation. To replenish its defenses, a cell pulls oxidized cysteine from the bloodstream using a membrane gateway called the System XC antiporter. This gateway operates as a revolving door. To import one molecule of cysteine, the cell must push one molecule of glutamate out into the extracellular fluid. Under massive oxidative stress, spinning this door constantly dumps huge amounts of glutamate, risking local neurotoxicity. Cellular survival depends on this precise, continuous supply chain of sulfur to neutralize oxidative threats before they can shred the cell wall. Observe what happens when this supply chain is exhausted by looking at a toxic acetaminophen overdose. A massive dose of paracetamol saturates the liver's normal clearance pathways, entirely forcing the drug down a secondary metabolic detour. This secondary pathway generates a highly reactive, destructive byproduct known as NAPQI. This chart represents the liver's total glutathione reserve. Under normal parameters, it neutralizes NAPQI toxicity. However, a toxic avalanche of NAPQI rapidly strips the entire reserve. Once the hepatic glutathione drops below 30% of baseline, the defense completely fails. Rogue NAPQI molecules now bind covalently directly to the vital mitochondrial enzymes of the hepatocytes themselves, triggering widespread cell death. The lethal outcome of acetaminophen poisoning stems from the rapid, localized exhaustion of hepatic cysteine, leaving the liver vulnerable to autolytic cell death. While a lack of cysteine destroys the liver, an inability to clear the oxidized form of the molecule targets the renal system. This occurs in cystinuria, a genetic mutation affecting the system B0+ transporter in the kidneys. Healthy kidneys act as a vacuum to salvage more than 99% of filtered cysteine back into the bloodstream. In cystinuria, that vacuum is broken. The kidneys filter the cysteine but cannot reabsorb it. Because cysteine is highly insoluble in acidic environments, and normal human urine typically sits below pH 6.5, so the molecule cannot stay dissolved. It precipitates out of the fluid, forming microscopic, flat hexagonal crystals. Over time, these dense geometric structures stack together, growing into obstructive urinary calculi. Because cysteine stones are faintly radio-opaque, they often appear as a smudge of ground glass on a standard abdominal radiograph, indistinguishable from routine calcium oxalate. If a clinician fails to perform microscopic evaluation for hexagonal geometry, they may miss the underlying transport defect until permanent renal damage occurs. A single broken transport gateway transforms an essential biological nutrient into an insoluble, organ-destroying mass. Measuring these delicate amino acid levels in a clinical setting introduces a complex logistical challenge for clinical pathology. How do you accurately quantify a molecule that physically alters its own structure the moment it is removed from the body's homeostatic control? The primary barrier for pathologists is the ex vivo autooxidation pitfall. This timeline tracks a drawn blood sample from minute 0 to minute 60. If the plastic tube simply sits at room temperature, the free cysteine instantly reacts with the oxygen in the air, binding to plasma proteins and disappearing from the measurable fluid. By the time the laboratory processes the sample, the results will display pseudo-hypocysteinemia. The patient's chart shows a false metabolic deficiency. The body is healthy. It was the blood inside the plastic tube that changed. Preventing this requires strict laboratory stewardship. Patients must fast overnight, blood must be drawn into pre-chilled tubes, plunged immediately into crushed ice, and centrifuged to separate the plasma within a strict 30-minute window. Even if the temperature is controlled, simply pulling a syringe plunger too aggressively can cause mechanical hemolysis, rupturing the patient's red blood cells and spilling their internal cysteine reserves into the sample, resulting in a falsely elevated reading. Without strict logistical discipline, the simple act of drawing blood will invent clinical emergencies that do not exist. To combat these pre-analytical errors, modern clinical platforms utilize automated software safeguards. If a patient's baseline free cysteine shifts by more than 10 micromoles per liter within a 24-hour window, a system delta check automatically flags the result, forcing a pathologist to investigate the sample's integrity. This chemical volatility also dictates how we treat an acetaminophen overdose. The gold standard antidote is N-acetylcysteine, or NAC. If the liver needs more cysteine to build glutathione, you might assume doctors could just hang a standard IV bag of the raw amino acid. They can't. Free cysteine oxidizes in the IV line almost immediately and relies on cellular transport gateways that operate too slowly to save the organ. By attaching an acetyl group to the molecule, NAC bypasses these limitations. This modification makes the drug stable in the bloodstream, allowing it to move directly through the hepatocyte membrane. Once inside, cellular enzymes strip away the acetyl group, instantly unpacking a massive dose of pure cysteine to rebuild the glutathione pool. Altering the molecular structure of the antidote is the only clinical method available to beat the clock and prevent complete organ necrosis. From the thermodynamic power of the covalent disulfide lock to the intracellular defense of the glutathione hub, from the mechanical failure of urinary solubility to the fragility of laboratory autooxidation, human architecture demands a precise balancing act. It utilizes a highly reactive sulfur core to form the bedrock of our physical strength and our ultimate defense against cellular collapse.